ApoE has almost all the risk, and I mentioned that a few moments ago when I was explaining ApoE. But it's a gene that we all have, and we all have it in 3 varieties, ApoE 2, 3, and 4. And if you have more of the 4 alleles, either 1 or 2, there's a higher chance of getting Alzheimer's disease. But it's not massive. There are many people who have Alzheimer's disease who don't have any ApoE4s. And there are people who have both E4s. Terrible, the worst terrible genotype, who don't get dementia even when they're 90 or 100. So it isn't a deterministic thing, it's just a risk. Then there's SORL1, which also seems to have some risk, maybe 5 or 10-fold lower. A few weeks ago, while I was here, the results of a big genetic-wide GWAS study showed that maybe clustering PCOM and CR1 might be involved, but it needs to be reproduced. Some of these others, less clear. What about Lewy body disease? I really have not been involved. I've been involved in some of the SORL1 and some of these others, but I'm not going to talk too much about that, about SORL1 and ApoE. Rather, I'm going to talk to you about a new risk factor gene specifically for Lewy body dementia, which I mentioned that Lewy body dementia was second in commonness to Alzheimer's disease, very common, that it overlaps with Alzheimer's disease, and that we're often not very good at telling it apart. Even though when it's really dramatic, we usually can tell, a lot of times we really can't tell, particularly for the first years. So this is an article from earlier this year that Lorraine Clark and Karen Martyr and myself and colleagues looked at the gene glucocerebrosidase, or GBA. Many of you may remember this from your biochemistry courses, because GBA is the gene that causes Gaucher disease, a well-known storage disease. Not so common here in Asia, but it's very common in some European populations, particularly Jewish European populations, interestingly, and also some Arabic populations. But it's also known as beta-glucosidase. It's on chromosome 1, and it is a glucosidase. It leaves a glucosidic linkage. And if it's completely absent, people get this bad multisystem disease that affects their liver and other parts of the body. It's a complicated gene. There's a pseudogene that's almost exactly the same. It's downstream and has complicated alternative splicing. But the interesting thing is that people have shown that GBA mutations seem to be increased in people with Parkinson's disease, and as we showed, in people with Lewy body dementia. So we found in a group of 187 brains a whole bunch of variants or mutations in the GBA gene. And these were all, almost all, heterozygous. They weren't homozygous. So these people did not have Gaucher's disease. Not mild, not moderate, not severe, not any Gaucher's. All they had was Lewy body disease. And so they had one mutation for the most part, and except for one compound heterozygote. And I don't think I'll go through every aspect of this study, but what I'll say is that there was a big increased risk from, it wasn't 100%, certainly many people with Lewy body disease did not have a GBA mutation, and a few people with GBA mutations did not have Lewy body disease. This is all pathology proven, so the good thing about this is, unlike the prior studies on Parkinson's disease, where they just thought the person had Parkinson's disease and weren't sure, here we know from the pathology of the brain that every one of these people who we call Lewy body disease had Lewy body disease, and everyone that we said did not have Lewy body disease did not, because it's only pathological material. We got the DNA from the brains. And so overall, regardless of whether we adjusted in regression analyses for age, sex, Alzheimer's disease, ApoE, dementia, no matter what we adjusted for, we got a rather strong risk of the GBA mutation for the presence of Lewy bodies in the brain. And you can see that we had no risk for Alzheimer's disease. If anything, there was a little bit, it wasn't, if anything went the other way, maybe a little bit less likely, but I'm not sure I believe that. So the GBA seemed to act as a risk factor for Alzheimer's disease. So I'll tell you a little bit more, that's published. I'm going to show you a little bit of unpublished work, which is that there are other lysosomal storage disorders. So let's say it's true. I mean, it looks true. Other people now have replicated this. In fact, even here in Japan, there's studies now on at least Parkinson's disease, which is similar to Lewy body disease, showing GBA increase. So let's say it's true. Well, why would GBA affect Lewy bodies? Why would it cause alpha-synuclein degeneration? Well, it may be because it has something to do with protein processing and lysosomal pathways. So we thought, well, maybe other lysosomal storage disorders, maybe some of these other genes that are sort of out of a medical textbook might also be involved in Lewy body disease. And this is very preliminary work that we presented in the spring, Lorraine Clark and myself and colleagues. On these 3 genes, we found that overall, the presence of one of those genes, here in the red, was associated with some Lewy bodies increase. Particularly interestingly, subcortical Lewy bodies kind of like Parkinson's disease, instead of the more diffuse Lewy bodies of Lewy body dementia. So the GBA was associated with Lewy bodies anywhere, particularly in the cortex. These other ones that were not GBA, but still another lysosomal storage disorder, seem to be associated with subcortical Lewy bodies. If we put them together, then we didn't get much. We just the GBA effect overwhelmed any effect of the other mutations. So we also looked at whether there was any interaction between GBA and ApoE4, and I won't go through this slide in detail, except to say that there did not seem to be any interaction. The way it seemed was that ApoE4 is a risk factor for Alzheimer's disease, that GBA seems to be almost complementary risk factor for Lewy body disease. And so this is a new concept, that before the only degenerative disease for which we had a risk factor for sporadic disease was ApoE4 in Alzheimer's. Now we think that there's enough evidence that maybe GBA is a risk factor for the second most common degenerative disease, Lewy body disease. And this is very preliminary, but it may be that other lysosomal storage disorder mutations, like I just said, in mutations like hexaminidase A and other genes, might also be a risk for Lewy body disorders. But we're not sure of that. That's very preliminary. If it's true that GBA and lysosomal storage disorder mutations somehow relate to risk for Lewy body disease, presumably that's because of some effect on protein processing, even in the heterozygous state. So even though a partial deficiency, not a full deficiency. And that could well be true. So in the last 5 minutes or so, I thought I would just quickly take you through, in Alzheimer's disease, where we are in terms of therapies. Because just as we think that protein processing of alpha-synuclein may be important in Lewy body disease, there's quite a bit of evidence that protein processing of amyloid is what's important in Alzheimer's disease. So there's a lot of different therapies for Alzheimer's disease, but none of them are all that good so far. In the past, all we had was non-specific therapies. If someone had hallucinations, we could try to reduce them. If someone couldn't sleep, we could try to make them sleep. If someone slept too much, we could try to make them sleep less. But that's all we could do. We couldn't affect the disease itself. Now, the last 15 years or so, we've had specific neurotransmitter-based therapies for Alzheimer's disease, one of which is a drug called donepezil, which I'll show you in a moment, that came from Japan, from Asing.